Good evening, everybody, and welcome to tonight's webinar. We're going to try and make it interesting. We're going to be talking about something that is become very topical, but not very well taught at medical school, which is the gut microbiome. So Kimberly sent me a WhatsApp to say, I hope you know you on camera. Yes, I do. Today, we wanted to make sure that there's no glitches because there's always glitches when I'm doing a presentation. So we wanted to make sure we get it set up quite early. And we had a practice round over my receptionist and my PA had a practice round to make sure everything was going to be fine. So let's start. Today's topic is the gut microbiome. And it's a very exciting topic because if you look at it, I think the next 10 to 15 years belong to two things in medicine. One is immunology, and the gut microbiome plays a very important role there. And the second is, of course, this decade belongs to the GLPs and all the new molecules that are going to come into the space combined with GLP to manage obesity, fatty liver, and probably some of the other inflammatory conditions as well. So these are my disclosures. So let's start by setting the scene because I think this is vitally important. And I'm just going to start off with something probably everybody in this audience is afraid to. So when the first case of HIV was ever found, the person who was examining the patient didn't know that the client had something called HIV. And obviously, these were some of the late manifestations of HIV that were being picked up. And it was only later that they found that there is something called the HIV virus, which caused this patient to present with AIDS, which is basically the AIDS it's basically what defines HIV. It's all the N presentations. So what I'm trying to get at is that now we know that that virus in that particular individual was around probably for at least a decade to 2 decades. So when they look at the HIV virus and they look at how quickly it's mutating, through that evolutionary pathway, you can actually go backwards and see when did this virus actually come into being from basically monkeys, when did it jump to humans? And it's predicted it probably happened in the late 50s or 60s. And you can do that by going backwards to see how fast the gene mutated. So the reason why I'm starting off with that is that all of us lived through a phase where there wasn't anything called HIV or AIDS, and then we saw what happened, and then we looked at the aftermath of what happened. So when the homeopaths first started talking about leaky gut and the gut microbiome, I'm going to start with the first two slides to show you why the medical world shoo-shooed it. So this slide looks at the pre-germ theory. So prior to the 19th century, there's something called the miasma theory. And the theory was that Western trained doctors, people that come from the line of Hippocrates, thought that disease was caused by bad air. This is basically your ancestors as doctors. From your occupational perspective, your ancestors believed that it was bad air that caused disease. And even long before that, Hippocrates, the Greeks, and the Romans, the Gallians, recognized that disease could be spread, but did not attribute it to microbes. They knew there's something that can spread, but they didn't know what the cause was. And so the humoral theory dominated for centuries. And that is the belief that there is an imbalance in bodily fluids, which end up causing diseases. So this is the same guys that were the fathers of modern day medicine. These were the theories before that. Then came the rise of the germ theory. Louis Pasteur, 1860s, showed that microbes are responsible for fermentation and spoilage of food, leading to the idea that it could possibly cause disease as well. Joseph Lister in 1867 introduced antiseptic surgery for the first time. So they were doing surgery for a long time before that, but for the first time, we had antiseptic surgery. Robert Koch, 1880s, developed the Koch's postulate, which says specific microorganisms cause specific diseases. Prior to that, it was bad air and an imbalance in fluid energy, bodily fluids that cause disease processes. So with the discovery of pathogenic bacteria, the idea emerged that the inside of the body, especially blood and internal organs, must be then sterile. Because think about it. Up till then, people were fine. They were walking around. And then this guy, Robert Koch, finds specific microorganisms that cause specific disease. So if I'm not diseased at the moment, that means there's probably nothing in me. And that means my organs must be sterile. And so then the widespread use of antiseptics and sterilization in theaters and wounds, and much later, we come up with antibiotics. So you need to understand why modern medicine is finding it so difficult to accept this whole gut microbiome issue, which the homeopaths have been talking about from the early 70s. It's now 50 years later where there's a bit of an adoption. And this is modern understanding. So scientists have discovered that the human microbiome, vast communities of bacteria, fungi, viruses, and archaea live in and on the body. So bacteria we all know about, fungi we all know about, fungi, viruses, and what's this archaea? So this is not a bacteria, it's not a virus, it's a unicellular organism, and its organelles are not surrounded by any membrane, and it is distinct from bacteria and fungi and viruses. So these things are living on us and in us. So today we believe that some organisms can actually be beneficial, challenging the idea that the body is sterile. So that's the onset. So let me tell you guys what convinced me otherwise very early in my career. So here's little Bana at Wits doing a BSc, didn't get into medicine because I messed around in my matric year, discovered girls and sports, but late in my life. And I ended up doing a BSc. And you guys all know the first year common subjects. And then I got accepted and they said, you take a gap year. And then I didn't take a gap year in that year. I carried on with second year BSc, found it interesting, and then ended up finishing my BSc. And my majors were genetics, human and animal physiology, but also I did microbiology. And so there, it wasn't theory, it was accepted at gospel. Our bodies were sterile, and anything outside of our body is potentially an enemy, and it can cause infections. And so from that perspective, what convinced me that there was something about modern medicine that we were missing was actually an illness. For those of you who know me, so I went backpacking through India when I was 21 for 65 days. And on day 17, I'm in Kashmir with 3 other friends. And to cut a long story short, we were very hungry. We were up in the mountains, basically not skiing, but I think what skiing was known as then in India. And we were very hungry. And there was absolutely no place you could buy food. And there was a guy making omelets on an open fire. I just came out from microbiology. And the guys are insisting we have to eat. And I said, guys, no street food. This is India. We're going to get sick. And they said, listen, man, just tell him to burn it a bit more. Anything there, it will die. And so anyway, we ended up eating those eggs. And whole and behold, we all four of us got salmonella poisoning. It was only a few days later when we see in the national newspapers that the Indian government was suing the biggest hatchery for letting out a lot of eggs. And there was a lot of salmonella poisoning going around at the time. Needless to say, I had 17 days of diarrhea. My weight was down into the late 40s, and we were still traveling. I think most of us, our luggages were heavier than we were. Now, where's the pearl in the story? We obviously carried antibiotics from South Africa in case, you can get the Delhi belly or the Bombay blues. And none of that shit worked. None of it worked. And we were really becoming sick. So anyway, in Delhi, through this family of the family friends of one of my friends who was traveling with us, directed us to this doctor called Dr. Aurora. And anyway, we went to go and see her at the Khan market, and she took interest in us because we looked Indian, but we had this accent that she wasn't familiar with. And she started talking to us. And anyway, when I related what happened to us, she said, listen, my boy, you look Indian, but your stomach is not Indian. And your antibiotics are not going to work here. And so what she did is she gave me a script and she wrote down 50 billion lactobacilli. And she said, go over to that pharmacy and you're going to get it there and start having this. And I almost died of shock. I said, what are you talking about? This is going to kill us. I mean, we already got an infection. Anyway, at the end, all the teaching in microbiology just went out of the window when you stick for so long and you're just throwing it up from both ends. And anyway, my friend said, you're going to be studying medicine, you take it first. Anyway, I took it, and within 24 hours, my diarrhea settled. And so that's for the first time I started understanding that modern medicine had some issues that we needed to deal with. So I needed to relay this because there's a lot of people in the field of medicine that have been shoo-shooing stuff that a few of us have been doing for the last 2 decades about the gut microbiome, but the time has come for all of us to embrace the new knowledge that is out there. So, what is the gut microbiome? The gut flora, the gut microbiota, microorganisms, which include all the things that I've just mentioned, bacteria, fungi, viruses, and archaea, that live in the digestive tracts of humans and other animals, including insects. The GIT tract and metagenome is the aggregate of all of the genomes of the gut microbiota. So I'll show you where that is going to. So the gut is the main location of the human microbiota. So the highest concentration of these foreign organisms, I shouldn't use the word foreign, basically in the gut. The second most colonized space in the body would then be the mouth. And then you find them elsewhere as well, and we're going to go through it in today's talk. So there are roughly 40 trillion bacterial cells. So you'll be hearing about people talking about there's hundreds of trillions of cells. They're right, cells. Here it's just 40 trillion bacterial cells. There's a whole lot of viruses, there's fungal material, and then the archaea as well. And in all of this, there's only 30 trillion human cells. So they outnumber us. So these trillions of microbes exist in the intestines and on your skin. And now I'm going to show you where else they've found them as well in the next few slides. Most of the microbes in your intestines are found in pockets of your large intestines. It's called the cecum, and this whole thing is referred to as the gut microbiome. The intestine is considered one of the most densely populated microbial habits basically on the planet. Now we're talking about the large intestines here. In an individual weighing 70 kilograms, The human gut microbiota gathers more than 100 trillion microorganisms and weighs up to 200 grams. The human genome, which all of you guys know, took a lot of the geneticists with, I think it's close to $5 billion US in 20 years to map out 23,000 genes. That's what's making up the human genome. The microbes within you encode for over 3 million genes. And so can you imagine the thousands upon thousands of metabolites that are being made by these living organisms in you and I? So where does this microbiota come from? So the first concepts were you're exposed to microbes when you pass through your mother's birth canal. However, new evidence suggests that babies may come into contact with some microbes even while they're in mom's womb. But for now, most people believe a former one, and therein lies the first problem. Because, and I'm the first one to say, when we first fell pregnant with our firstborn, and the lovely gynae who delivered my boy is in the audience today, and we're all ever thankful to her. We just knew we were pregnant. We just did the first scope, and she said, Sandeep, when do you guys want to do the Caesar? It was like a no-brainer. Any medical person would want to go for it, right? I mean, you don't want stress incontinence later, and you don't want to be going through the hassles of quite a few things that can come with a prolonged vaginal delivery. However, therein lies the first problem, because if you don't come through your mother's vaginal canal, the bacteria that's going to colonize you is going to be the surface bacteria that come from the skin, as opposed to the vaginal vault. And that's why you're going to have a completely different microbiome than what you were evolved to have. So although there is a core consisting of bacterial groups common to all healthy humans, and it's estimated that about 1/3 of the gut microbiome is common to most humans, the other 2/3 will vary. And that each person has a composition that is uniquely theirs, almost as unique as your fingerprint. But what are some of the other factors other than your mother's vaginal vault or skin microbes that govern what yours is going to end up looking like? So feeding becomes important. So that's where breast milk will trump artificial milk. And then once you start introducing solid foods, the type of solid foods, whether it's processed or not, whether it's freshly made, whether it's from a packet, whether it's from purity, and so forth, will influence the gut microbiome. We know antibiotics affect it. So if the child's had an antibiotic within the first 3 months of their lives, it's going to change the microbiome completely. And so we do know prem babies now being born, you can have meconium issues, and there are needs for some babies to be going onto antibiotics. So that will definitely affect it. When you're an adult, suppressing the acid production in your stomach is important. I mean, there's a reason why every living species has evolved to have, if they've got a stomach, to have acid in the stomach. It's there to help digest proteins. It's there to help guard the fort from allowing bad bacteria to colonize the lower part of the gut. Antidiabetic drugs, psychiatric drugs. Anyway, there's a lot of things that can affect the gut microbiome. Dietary habits. So the type of foods we eat, the way the food is prepared, processed food, foods with preservatives, all of that influences the gut microbiome. The environment, very important. So somebody living close to the equator is going to have a better gut microbiome than somebody in Norway or in South Africa. So as you move away from the equator, the risk of type 1 diabetes has been shown to increase. Type 1 diabetes is an autoimmune condition. And so as you move away from the equator, the diversity of the bacteria or the living organisms, and especially the parasites you're going to be exposed to while growing up, is going to be different. That's why children that have worms are unlikely or never going to develop asthma or allergies. And so, yeah, with studying what's happening with autoimmune conditions, we're starting to better understand the involvement of the gut microbiome in all of this. Somebody living in a rural area compared to urban is going to have a different gut microbiome. In the urban areas, we're a lot more sterilized. And weight in itself will influence the gut microbiome and vice versa. So my patients always ask me, but why do I have this? And I tell them, just go and watch 1 or 2 TED Talks on the gut microbiome, and you'll understand that there's no way your doctor is going to be able to know when this problem started. Of course, I'm unique in the sense that I did study. I had a medical background, and for me, it was easy. I had that event, salmonella poisoning, and I knew ever since then my gut wasn't right. And so your gut microbiome composition differs from area to area. So if you look to the top right, these 5 types of bacteria dominate the mouth. Firmicutes, Proteobacteria, Bacteroides, Actinobacteria, and Fusobacteria. If you look on the bottom right, in terms of what's happening in most people's healthy gut microbiome, Actinobacteria, Bacteroides, Firmicutes again, Lactobacilli, Streptococci, and certain Enterobacter. If you look in the lung, different types of species dominate. And if you look on the skin, different ones. And now look at the vagina. So a child born through the vaginal canal is going to be soaked and swallow a lot of lactobacilli. Whereas somebody born through Caesar are going to have the other 5 dominating. And so and that's why people say 2 things. One is, if you're going to have a Caesar, make sure mom's awake. So there's no anesthetic effect. Number 2, try and make the Caesar cut very small, almost mimicking what's happening with mom's vaginal canal. And then between the obstetrician and the assistant, you're going to try and get the baby to actually be delivered with through the canal. Try and get the neck and the back to basically go through that arch as well, for future importance in terms of what's going to happen to the spine and posture. And then this is going to sound disgusting to some of you new to the audience in terms of gut microbiome. Take your swabs, rub mom's vagina, and then actually rub it onto the child. And that gives the child a better chance from an immune perspective. And then, of course, the more breast milk, the better. So when this slide was made, and it's not my slide, that is what was known in terms of what was colonizable. And then we come to the gut brain microbiota. So recently, about 3 or 4 months ago, they found that the brain is not as sterile as we thought. We thought this blood-brain barrier prevented anything from coming through and everything was very selective. Well, now that's bullshit, because they found living organisms in the brain. Now, the gut-brain connection is very important, because the type of food we eat influences our neurotransmitters. The type of bacteria we colonized with, and that can vary from time to time in terms of which are dominant species, will affect cognitive function, will affect your happiness factor, sorry about the spelling there, and even influence your social interactions. There's been links in terms of stress and anxiety, and then even links with basically neurodegenerative conditions like Parkinson's. So on the left, you can see all the different things that can influence the gut microbiome. And on the right, you can see how it's influencing our behavior. So one needs to question who's actually in charge. So what we now also know is that the immune system is probably the most profoundly affected by the gut microbiome. In fact, they probably work hand in hand. So the GIT tract hosts a large number of immune cells. In fact, it's estimated, depending on what studies you read, 70 to 90% of your entire immune system is concentrated in the gut area, basically from the mouth down to the anus. And so the GIT tract hosts a large number of immune cells which constantly communicate with the gut microbiota. The maturation of the immune system needs the development of these commensal microorganisms. One of the mechanisms of the gut microbiome affecting the immune system is by mediating neutrophil migration, which will subsequently impact T cell differentiations. So the neutrophil will eventually affect these two very important cell lines with immunity, which is T helper cells and T regulatory cells. Disorders in the microbiota development during the maturation of the immune system could lead to deterioration and immunological intolerances. So here it's immune tolerance and autoimmune diseases. So what happens is when you end up having a leaky gut, which we're going to go through now, it's going to end up causing you to have intolerances, which can eventually lead to allergies. But it's first an intolerance before it becomes a memory cell. And because of this leakiness and through something called genetic mimicry, you start having autoimmune diseases as well. And so additionally, heterogeneous molecules produced by microbiota may induce immune response and stimulate inflammation and chronic tissue damage. So I'm going to touch on this a bit later in terms of the importance of postbiotics as well. But here's a cartoon of what looks like a good gut. And so what's a good gut? There's the lumen. So if you've got the gut, for the non-medical people in the audience, so it's like a hose pipe. And the empty part inside is where the food you eat, the water you drink, and the fluids you drink go in. And basically, at the end, it's going to come out of the back passage. So if you had to just elongate yourself, you one long tube. And along this tube is a mucus layer that is actually smaller than the length of one cell. Now, I'll tell you why that is important, because that is the single most important part of this discussion. So in that layer lies bacteria, other living organisms, and that is the first lead to communication between the outside world and the inside world. So between those 100 trillion organisms and the food you ate and the heavy metals that came through and the insecticides and herbicides and pesticides that the farmer used in the food and all the preservatives that our food industry puts in there and the colorants and the stabilizers, you name it, flocculants, there's so many things out there. All of that, there's only one layer of cell between the outside world and you. Whereas if you look at your skin, there's quite a thick layer protecting you from what's happening from outside. And that's why, can you understand why 70 to 90% of your entire immune process, immune system is concentrating in the gut? Now, there are tight junctions between these cells to prevent stuff from coming through. In this layer is happening a lot of signaling. These short-chain fatty acids are produced by certain bacteria. And if this one produces butyrate, it helps that one survive. That one's waste product makes this guy survive. This guy's waste product makes that guy survive, and so to speak. It's a very strong symbiotic relationship between these 100 trillion organisms, and that balance is vitally important. And that signaling is what's modulating the T regulatory cells, the helper cells, so what you call helper and suppressor cells. And that is what's maintaining immunity. This barrier is impermeable. So you've got one, I reiterate, one single layer of cell between you and the world. And when you've got a disease process, when there is an imbalance in this microbiota, overgrowth of some species, it happens at the expense of the others not growing. And certain species then produce LPS, they're producing toxins, they're producing waste products that are not conducive to health, like these short-chain free fatty acids. You start having that mucus layer disappearing. And when that mucus layer disappears and you have dysfunction at the barrier, you start having these tight junctions opening up, and you have bacterial or anything else that can come through, macromolecules from coming through. Remember, when you break down fat, at the end, it all comes down to triglyceride. When you're eating any protein, it gets broken down, and then you get a whole lot of amino acids, and you get one or two amino acids attached to each other. And when you eat any milk or dairy products or carbohydrates, they get broken down into single molecules or just two molecules. And that is easily going to be absorbed through the brush border. However, when you've got an opening in the tight junctions, you're getting larger macromolecules passing through, which is going to be making it more antigenic, and anything large, foreign, your immune system is going to be producing an army of cells to fight that. And the problem is through genetic mimicry or molecular mimicry, that antibody that you produced against that guy or this guy now starts attacking the thyroid, because the thyroid tissue through genetic or molecular mimicry is looking a bit like this guy. And in another patient, the antibody is attacking this guy and then mistakenly attacks the joints, causing rheumatoid arthritis. And in somebody else, that one, the antibodies are produced against that guy, and then through genetic mimicry, it goes and attacks the cells of the skin and causes depigmentation and ends up with vitiligo. So autoimmune conditions are caused by your own cells going array. So it's basically attacking its own body because your tissues are looking like something that it experienced from this leaky gut. So this cartoon shows it a bit better in terms of what we mean by the tight junction. And then you got a whole lot of good bacteria, good mucus layer, healthy environment. And now here, with bad bacteria, with gluten, with toxins, the tight junctions open up and these guys go through. And that's why the homeopaths coined the term leaky, because that's exactly what it is. It's allowing things to come through that's not meant to come through. The modern term is dysbiosis, but this one explains it a bit better, but you can use whatever term you want to. So what's the role of microbiota then in the development of disease? And I've touched on it. So this microbiota, which is a complex system consisting of trillions of microorganisms with advanced sequencing technologies and bioinformatics, these are big words. Most of the microbiota related research is focusing on the relationship between these organisms, compositional changes, and various disease states. Now, a few years ago, a decade ago, we thought, it's going to be easy to study these guys because, listen, we just take 1 million people with asthma, check what their gut microbiome's like, and compare it to 10 million people with no asthma and see what's the difference. Makes sense? Do the same with diabetes. Do the same with vitiligo. And you're able to identify the bug. So what went wrong with those kind of studies? Well, it's almost like quantum physics. I don't know if any of you guys are interested in quantum physics, but there's a principle in quantum physics is that when the observer tries to look at what's happening, the observer changes the outcome. And so it's the same thing here. When the observer went and pulled these microbes out of the environment to study them, they were no longer representative of what was in that gut, because most of these guys are living in an anaerobic environment. And so as soon as you pull them out, they die. That means you can't grow them on a micro dish. And then this organism's waste made this guy survive. And this guy's waste made that one survive, and so forth. So the minute you start trying to culture this guy and he's dead, it influences everything else. And then they realize they're back to square one. They're not going to be making as much money as they thought they were going to. Because the pharmaceutical race for basically conquering the gut microbiome started well about a decade and a half ago. So then came the clever guys, the geneticists, and they say, okay, what the hell? If we can't identify what the hell this guy is doing, their DNA is all different. Let's just nutri-bullet all of them and let's study the DNA. And through the DNA, we'll be able to know which species are there. And using that in bioinformatics, you then map out the person's gut microbiome. So when subjected to external changes, the balance of the microbiota community can be affected, leading to dysregulation of bodily function and disease. So even when we take them out of the environment, they're difficult to study. And basically a lot of external changes, when they affect the imbalance, it's going to affect bodily function and disease. And what kind of diseases can you get? Cardiovascular disease, cancers, respiratory diseases, diabetes, irritable bowel disease, irritable bowel syndrome, brain disorders, chronic kidney disease, and my big one, the liver disease. For years, I've been treating patients with fatty liver by trying to fix the gut microbiome, because we had insulin resistance for long now. I mentioned this at my last talk. I mean, at Barra, we've got tons of insulin-resistant patients, and yet we could never recruit a patient for any of the fatty liver clinical studies. And I was always wondering why, until we started working on the gut microbiome. And I think it's a dual thing. When you're sitting with a leaky gut, remember, whatever's been absorbed, the first organ to face the shit is the liver. And so if you take that and insulin resistance, you've got the potential of liver issues. And so here the gut microbiome has been shown to affect liver disease. And so most of the studies are basically on bacteria, because there's very limited study on the non-bacterial species. That is a lot more difficult to discover. So this is still in its infancy. We're not even employing the use of viruses, the phages, the fungi. I mean, the mycelium plays such an important role on Earth. I mean, the mycelium extends from here right to Pretoria. I mean, if you go into the ground, and it traverses continents, and these things are communicating with each other. So the fungi are probably going to be playing a very important role with us as well. I mean, just speak to the guys who've taken a mushroom trip. And of course, these archaea, nobody even knows what they're all about. So disruption of the gut microbiome, leaky gut, allowing things to come through, causing an immune disruption. What does it lead to? It can cause heart disease, contribute towards heart disease, causes inflammatory bowel disease, liver issues, kidney, brain disorders, diabetes, respiratory and allergies, and then cancers as well. And so it's an interplay between the metabolites, which are the vitamins these guys produce, which are the postbiotics, the free fatty acids, the short chain fatty acids, and then the immune modulation, interleukins, tumor necrosis factors. And so it's an interplay between these inflammatory and non-inflammatory issues. So for those of you who are still not convinced that the gut microbiome plays an important role. These are studies not done by me or any of you. These are clinical trials being done all over the world in different phases, phase 1, phase 1b, 2, 3, 4, in the field of allergy, cancers, diabetes, C. diff, heart disease, HIV, inflammatory bowel disease, mental disease, neurological conditions, obesity, bowel disease, infections, inflammatory conditions, and all over the world. There's tons of research taking place. Some with response, some with no response. Like with any clinical trial, some things are going to work, some things are not going to work. So best you start becoming au fait with the gut microbiome. This is another slide from another site, taking a bigger look and in a bit of more details in terms of how the gut microbiome influences all of these conditions. And it's not for me to go through each one, but you can see cancers, autoimmune, the metabolic conditions, autism, psychological things, mood, schizophrenia. It's all there, guys. There's nothing that the gut microbiome doesn't touch. So let's take a more detailed look at the gut microbiome and its physiological function. Let's look at digestion and nutrient absorption. The gut microbiome helps break down complex carbohydrates and other nutrients, proteins and fats, which humans by themselves cannot digest. So we need these guys to help us break down our foods so that we can start absorbing stuff a bit better, because a lot of stuff we're not going to be able to digest ourselves. I don't know if you guys know that there are bacteria that produce vitamin Bs, and that a big source of our vitamin Bs are actually not only from the foods from outside, but these bacteria manufacture it. In fact, B12 is very much involved in this pathway, and vitamin K. Remember, without vitamin K, you don't clot too well. If you don't clot too well, you bleed to death. From an evolutionary perspective, it was very important for you not to bleed. And so these guys are very much involved in the processes that we require to survive. Let me remind you that without vitamin B12, your DNA cannot split, replicate itself, divide, and make one cell into two. The entire DNA replication is very much dependent on B12. So it is not just about digesting nutrients or energy. There's a lot happening here. These short chain fatty acids, like butyrate, acetic acid, propionic acid, and there's a few others, are a very important energy source for your gut cells. That one single layer of gut cell relies on these guys to survive. The mucus layer, very much important, very much reliant on these short chain free fatty acids. And so their waste products will influence gut motility, gut absorption, secretions from the gut, and has a big impact on digestion. So the gut, bit of a no-brainer. Immune system modulation. The gut microbiome is a critical component in the immune system. Its role is in the training and modulating of immune cells. It helps prevent the overgrowth of harmful bacteria, and it can stimulate the production of antimicrobial compounds, which are, of course, very important for your defense. The gut microbiome also influences the development and function of lymphoid tissues in the gut. I don't know if you know it, but the entire gut got Peyer's patch and lymphoid tissue, which is important in terms of immune response. That's where you start recognizing in your lymphatic system what is self, what is not self, and you're making immunoglobulins and a lot of other things that are important for you to recognize as what is important to attack early on in that infection. What about metabolic regulation? The gut microbiome can influence the host metabolism by producing metabolites that impact energy balance, glucose homeostasis, and lipid metabolism. Let me expand this a bit for those of you, and I think I've mentioned it on one of these webinars last year, that years ago we used to, yeah, I did. We spoke about obesity, okay, on this platform. So years ago we used to think energy intake, you're taking 500 calories, and basically it's an energy input and output. So if you don't burn it, you're going to put on weight. And that's why people say just eat right and exercise and all will be fine, which is very important. But obviously, that's not the whole truth. So they found that if you and I, let's say the same height, same gender, eat a meal, same amount of calories, that from one individual to the other, the absorption of calories can vary up to 30%. That's a lot. So what influences that? The gut microbiome. They've shown that certain gut bacteria will break down your calories from your food and allow you to absorb more calories than another person having eaten the same meal. So whilst you think calorie input and output the same, is all of it being absorbed or not? And in some people, you might consume 500, but 250 might pass out the other back passage and you'll absorb 250, whereas in somebody else, same meal, a lot more gets absorbed. So they have a strong impact in terms of this energy balance. Did I ever tell you guys about the rat study, the OB mice and gut microbiome? Let me just digress a bit because it's important for you guys to understand this role in terms of calorie absorption. So years ago, they did this elegant study. They took OB mice. OB mice are mice that are genetically bred to become fat and become diabetic, so you can study your diabetic drugs on them. And so I'm not doing these, by the way. I just read the article and I'm telling you guys about it. So none of the tree huggers like my wife must come and attack me for the study. So you've got the same litter from the same mom. So the genetics are the same. You've got the same gender. So two male mice. And they kept it in the same lab. That means same environmental temperatures. Same food. It's called chow to make them diabetic. And you know it's cruel, but you can exercise them to have equal energy outputs. So you can make them go on treadmills, and there's ways of getting them on the treadmills, which I'm not going to discuss here now. So basically, it's a very well-designed study. Can you see? You've accounted for the genetics, you've accounted for gender, you accounted for input of calories, you accounting for output in terms of energy expenditure, and then the environment, which we know pollution and air temperature and all of that is controlled because they're all in the same cage, right? Divided by this perspex stuff. And so one would assume that both will become fat at the same time and become diabetic. All well and fine in theory. So what they did was they did that. And so, yeah, these guys just grew and they're ready to be diabetic. But then what they did was they had one mouse sitting in another cage, not from the same genetic litter. And that guy was a bit thinner, lean. So they collected that one's stool and inoculated one of these brothers with that stool. And they still continue with the same experiment. And all of a sudden, this guy loses weight with the same amount of food being given, same environment, same genetics. And remember, he became fat prior to this on that same chow, and now he lost weight. So therein was the first clue that the gut microbiome is influencing what's happening with energy absorption. That's not where the experiment ended. So what they did was they kept the stool of one of the big guys and gave it to the genetically different thin mouse. And that guy became fat. And the best part about the study is they kept that stool and gave it to the mouse that was fat and became thin, and he became fat again. So can you see where stool inoculation is going to be going in the future? People are going to be trading stool. And I tell you, it's going to become a business. So from that perspective, that's where the thought of some people, you'll go and YouTube it, you'll see they want to actually go for fecal transplants for different reasons. And I'll tell you why we haven't offered it all these years when the temptation was there to do so. Okay, so it can also affect the production and release of gut hormones, which play a role in regulating appetite, insulin sensitivity, and other metabolic processes. And so this impact on absorption and metabolism of certain nutrients will influence obesity, diabetes, and in fact, all metabolic condition. So definitely playing an important role in metabolic condition. What about other diseases? So it's been linked to a wide range of stuff. We know inflammatory bowel disease was right early on. So the disease is different to the syndrome. The disease is where you can prove inflammation. You know, you've got ulcerative colitis, you've got Crohn's, you've got something in between. And irritable bowel syndrome is basically you feeling all the symptoms of bloating, loose, broken stools, or constipation, feeling uncomfortable. But when you actually go in, you're not seeing inflammation. And then, of course, it will affect, I just spoke about these as well. Okay. So now, this gut-brain axis that I spoke about. So the gut microbiome is also connected to the brain through the gut-brain axis, which is that's important, directional communication pathway between the gut and the nervous system. These gut microbiomes can produce neurotransmitters. So I'm sure some of you in this audience already know that actually 90 to 95% of serotonin, your happy hormone, is made in the gut. That only 5% is actually produced in the brain. And then, so most of you guys who have been referring patients to me or vice versa know that I do serotonin levels in blood, and I've been heavily criticized by psychiatrists to say, well, tell your doctor the serotonin in the blood is not the same in the brain. Well, excuse me, this is intraplatelet serotonin that's being measured at the lab. Platelets cross the blood-brain barrier from the last time I remember, because that's how patients develop stroke. They form a clot. You either have a bleed or a clot. So if the platelets were not making through the blood-brain barrier, then you wouldn't able to clot in the brain. And so, and if you know physiology, you'll know that within 30 seconds, the blood that was pooled in your forearm is in your brain. And so from that perspective, in 30 seconds, your serotonin level, where the reference ranges are 1200 to 6800, the average being about 3,500, is not going to magically go from 50 to 3,500 in less than 20 seconds or 30 seconds from the periphery to the brain. So whilst it's not an exact measure of what's in your CSF, it's the closest one you've got, because it's less in terms of playing darts than what's currently being done. Because nothing has been measured. It's just about symptoms, isn't it? Which is important, but this at least you've got to measure. And so here we know that that happy hormone, and most of its work is being done in the brain, is actually produced in the periphery. And so that can be influenced through the gut microbiome, and that's been investigated, and they've already identified bugs that produce the happy hormone. The gut microbiome also plays a role in development and maturation of intestinal epithelium and immune system. We spoke about that. They influence in the production of that mucus that lines the gut. And then, of course, its influence on the liver and kidneys because of that. So, how may we use this in disease treatment? So, there's the thought of, well, the food may play an important role. Prebiotics, the stuff that keep the good guys alive. I tell people, just like I got to feed my snakes, my tortoise, and my cats and my fish every day for lunch. I think even my staff are tired at looking at my lunch. I have a bowl of salad, which is 90% cabbage. Fortunately, it doesn't bloat me, so I'm feeding my pets in my gut microbiome. And then postbiotics as well, which are very important. Then there's probiotics, and I'm going to show you not all probiotics are equal. This fecal microbiota transplantation. So we do it with C. diff, and you're going to see they're going to be starting to use it for a lot more other conditions. Let me tell you now why I haven't done it for years and years and years. I mean, Prof. Ali and I and Bilal Bobat have had these discussions, I think over 15 years ago, even before the first study that came out from the Netherlands doing fecal transplant in 12 patients with severe lupus, and 6 of them got cured and 6 of them had a problem. And years before that, I was trying to convince Prof. Ali, who's the head of GIT at the time, to why don't we pursue in doing this study. And he just thought that the gut microbiome wasn't a big thing those days. And now he's fully into it. And Bilal was still a student, and now he's the head of the liver transplant unit. And I was trying to convince him to do his PhD on this thing. And by then, I was going quite fine, my practice in terms of trying to manipulate the gut microbiome. And finally, they at an international conference, and somebody presents a paper in terms of what they're planning to do. They haven't done it still. And they sent me a WhatsApp and they said, listen, we can beat these guys to it. We're up to it. We want to do your study. And when they came back, we had a discussion. And after using some very flowery language on me, Prof. Ali said, it took you so long to convince me to do it, and now you don't want to do it. And my problem at the time was that the ethics committees were only allowing you to use a stool sample from a younger family member in your own family. Now, does anybody want to unmute and tell me what the problem with that is? Just hazard a guess. Anybody, don't be shy. So remember, genetics play a very important role. We spoke about genetic mimicry. For you to have an autoimmune condition, you must have the genetic predisposition for that, and the gut oil becomes the second hit. And by then already I could see how this thing was influencing my patient's weight, and in terms of what was happening to their allergies and their reflux, and what was happening to their moods as well. So I got thinking, and I'm saying, shit, I wanted to use another word, by the way. I don't want to be now fixing somebody's problem X and introducing Y. I mean, the last thing you need is to fix somebody's weight problem and make them bloody schizophrenic, or fix them up from improve their irritable bowel syndrome and end up causing vitiligo, and or help them fix lupus up, and then you end up getting rheumatoid arthritis. You're just exchanging one condition for another. And so that's when I realized that getting the right person's stool is important, because although you may be doing a fecal transplant to get somebody fat to being thin, inevitably you might end up causing them to be depressed, or got lupus, or influence their cardiovascular risk, or even the cancer risk, because you take the whole package with you. And so that's why that needs to be much better studied. So we couldn't use a family member stool sample because that was already a problem stool to start off with. And I'll tell you what the thinking behind the ethics committees was, because you can see how rudimentary the thoughts were early days with everybody. Is they thought, hold on, let's use a young person's stool, assuming young people are healthy. They haven't played the poker hand out. On the contrary, I will choose a non-biological donor who is as old as time, who's got a good lineage, family history with no major autoimmune conditions or cancers or metabolic condition, who themselves have a stable mood, no major cardiovascular risk factors, because you can check their homocysteine, their fibrinogen, their cholesterol, their lipoproteins, their insulin-resistant markers. You can check all of that. You can check for as many cancer markers as possible, and rather take that person's stool, because they've already played their poker hand out, not the young guy who's still got 60 years to show you that they're going to get a cancer. So that's why we never embarked on that. And the long and short is, the guys in Netherlands did beat us to the study. And they came back the following year's conference and they said, you see, we should have done it. We would have been the first because 6 people got cured with the lupus and the other 6, the symptoms improved. And then in the years to follow, those same people that were studied were found to have other conditions that were worse than the primary condition. So that's why it's not just as easy as the patient who's got a BMI of 48 going to get a thin female patient's BMI of 22, just do a fecal transplant and hope for the best. There's a lot of other shit that can go wrong. Phage therapy is going to be huge. Engineered bacteria, huge. And then live biotherapeutic products as well. So anyway, let's touch on this. Now, what do you guys see here? Lots of different looking hojas. And biodiversity is the name of the game. My patients tell me, but why do I need so many? Why can't I just take my Rutary 1 billion or my Probiflora from Dischem? It's so much cheaper. And they say it's the 9 strain. Okay, this is why. If your favorite, this is what I tell them, if your favorite animal is a lion and you go to the Johannesburg Zoo and you realize that every cage only got a lion, by the third one you're going to be bored stiff, although it's your favorite animal. Biodiversity is very important. And the more numbers you got, and the more you got of them, the better balance they're going to have. And so we don't even know all of the ones that exist. We know there's 100 trillion, more or less. These are just guessed numbers. But we're still at the infancy of this game. So biodiversity is key, guys. How did cavemen do it? How did they do it 100 years ago? So you notice all old nations have a fermented item on the plate every day. I'm Hindu, so if you look at it in India, we have something called raita or curry, and that's either one of them will be presented to you every day, lunchtime, with your meal. And then there's lassi. I see, yeah, they got it here. You go to South America, the women there will chew the food, spit it out in the plate. I know it sounded in a bowl, and then they make a fermented drink. And so all the mothers of the village spit into this thing. And so imagine how rich this microbiome is, pre-digested with amylase, lipase, and all of that stuff. And then they let it ferment, and then you have an inoculate of this every day. You go to Scandinavia, and they've got fermented fish. You go to the Far East, and they've got tempeh, they've got miso, kimchi, there's sauerkraut, there's kombucha, fermented veggies, which is different to pickle, huh? Sourdough. Not all sourdoughs are equal. They need to be made with wild yeast, not a single strain. Yogurt, kefir, cheese, aged cheese. So there's a lot here that will aid digestion. Now, don't make the mistake of thinking miso or buttermilk is going to cure your inflammatory bowel disease. Remember, these were daily items that you took every day to maintain a good gut microbiome. Once you got a shitty microbiome, it's very difficult to put the burden on this kombucha, which a liter of it will give you 2.5 billion lactobacillus to end up trying to fix up a problem that is just far gone. So these are important to aid digestion and maintain gut health. That's why you need higher doses of stuff once you've already got a condition to overcome the problem. Once you overcome the problem, and which you can in most but not all for now, then you can maintain it with that. What are prebiotics? Just like your cat needs food, your good bacteria need food. These guys have fibers, different fibers that feed different bacteria. to keep that biodiversity going. So when you liquidize your smoothie in the morning as opposed to Nutri-Bullet it, there's a big difference. Big difference. So you want as much of the fiber in there to keep the good guys alive. The probiotics we spoke about, and then postbiotics. Some of these bacteria, the good ones, produce waste products that I spoke about, butyrate, acetic acid, propionate, that is very important for other microbes, but also for your single layer of cell that's protecting you from those 100 trillion different living organisms and foreign particles. Also important for the mucus layer. And also important for the inflammatory process within your body, because we also absorb those things. So these can help with postbiotics. So another picture from another side, just giving you same picture. And then what about minerals? Very important. So calcium increases gastric acid. 60% of people in the northern suburbs are on something to switch their acids off. I mean, how can that be right, guys? Think about it. For millions of years of evolution, it took for you to get to a point where the stomach is producing acid. That's like having a trellis door at home and leaving it open at night. What's the point? And so, of course, in the long term, it's going to affect the gut microbiome. Sure, short-term use is okay. I get patients that have been on it for 15 years. So how are you expecting to digest your proteins? And so calcium increases hydrochloric gastric acid secretion. It improves intestinal permeability and eases diarrhea as well. Magnesium controls over 300 things, guys. Facilitates calcium stimulatory function on the gut. Is important for regular movements of the gut. Important for gallbladder emptying, and then relieves constipation as well. Selenium, very important from an immune system perspective. Anti-inflammatory selenoproteins, very important. And it promotes microbiome balance. Zinc, okay, became very fashionable during COVID. Important for many functions. I'm not going to go through each. Electrolytes, important. Iron, where do you think iron's absorbed? In the gut. Too much is no good, too little is no good. Remember, there's a balance. So eating the right foods and making sure that the foods provide the kind of nutrients that you need. So what do people out there, your clients, think? What people think probiotics do? They help with stomach problems and reintroduce bacteria after antibiotics. True. But it's more than that. Real antibiotics should also reduce allergies, improve metabolism, improve skin. The dermatologists are becoming very big about the skin microbiome now, because for years they've been using high doses and long-term use of antibiotics. Surely that's not how these things were invented, and it wasn't invented to be used for such a long period of time. Surely you're going to affect the gut microbiome at the expense of skin microbiome. So now we're learning a lot more about that as well. It improves mood, cravings. It's supposed to prevent antibiotic-induced bacterial imbalances. It must drive out other health problems that we spoke about, improve lean body mass, increase absorption of healthy food. So it does a lot more. So where did this thought of it just helping stomach problems come from? Well, the first studies that showed that the bacteria might be playing an important role was on inflammation. When you had inflammation in the gut, the Lactobacillus plantarum 299, you guys recognize it, LP299. That was for inflammation. And then for the mothers out there who were giving your children Rutary, it was to dampen inflammation when you had a prem baby. And so then everybody thought these are the best. They were the best from the first lot that was studied, and they are the best. They're good for inflammation, not for something else. So let's bust some myths there. All probiotics are the same? No. That's like saying all antibiotics are the same. That's like saying all the vitamins are the same. That's like saying all the antihypertensives are the same. No, they all work differently. Probiotics are only beneficial for the digestive health. Yes, digestive health and many other things. You can get enough probiotics from your yogurt. Woolworths Bulgarian yogurt, the strongest. You have to eat that tub or that helping to get 250 million. That's very good to maintain good health, not to fix the problem. Probiotics are not necessary if you eat a healthy diet. Depends. Depends. So it depends where you are. If I've got Crohn's disease, I can eat as much of healthy food as I want to. It may just dampen my symptoms, but not going to cure my issue. All probiotic supplements are equal quality. No. Refrigerated probiotics are better than non-refrigerated ones. No. So certain gut bacteria, like Akkermansia, like Faecalibacterium pronicii, and those producing butyrate are linked to improving diabetes. How? By promoting insulin sensitivity, reducing inflammation, and enhancing glucose metabolism. Now, Akkermansia has become big. Beware. If your probiotic has Akkermansia and it's cheap, throw it away. Because just go and ask the guys who are making probiotics what 1 kilogram of Akkermansia cost. It is in the hundreds of thousands. And the studies have shown you need at least 50 to 60 billion to influence sugar or weight. There's a product coming out in South Africa that is being held by Sapkra with 10 billion. There's one coming out from this range with 60 billion. So it's the numbers game. It's a numbers game. That's what's important. Look at your bottle. Because they'll show you the study, and then you don't read the fine print. And the fine print said you needed 60 billion to get at least 3 to 6 kilogram weight loss in 6 months. Back to diabetes. The bifidobacteria species are the ones that are influencing insulin sensitivity and reducing blood glucose. The Coprochus influences insulin sensitivity. The butyrate producing ones, Rosburia and this Faecalibacterium, through its production of butyrate, has a protective effect on the intestinal barrier. Again, reduces inflammation, and this guy also improves glucose metabolism. Lactobacilli, which a lot of you know, that's the common acidophilus, the Gasseri type, and the salivarius type, also helps improve glycemic control and insulin sensitivity. This is saying the same thing, showing you how Akkermansia plays a role in terms of processing glucose as well. That's what's different there. So, and Akkermansia helps improve metabolic endotoxemia, which is a condition where toxins from the gut bacteria enter the bloodstream and cause problems. So here, inflammation is the key word. Metabolism of glucose is important, and the influences you're going to have through your short chain fatty acids on inflammation and on insulin sensitivity becomes important in diabetes. So can you see in the near future, there's going to be a concoction of bacteria, microorganisms, that's going to influence your HbA1c. About gut and asthma or gut and allergies. So while there's no single bacteria now directly showing cause effect to asthma, an imbalance in the gut microbiome, including a decrease in Akkermansia, Faecali, Bifido, causes higher risk of asthma, and an increase in Bacteroides and Streptococcus are likely to also increase asthma. So deficiency of these three and a higher amount of these will worsen atopy, allergies, and asthma. I mean, we get patients with wheals, or we get patients with hypersensitivity to something, and every 2-3 days they in an anaphylactic shock, or they get, why would they come and see an endo? It's only because we work in the gut microbiome, because you need to sort out the gut microbiome to decrease that, because no amount of antihistamines or cortisone will then able to sort that out. You have to get that gut microbiome right. What about rheumatoid arthritis? Research suggests that an overabundance of one of the groups called Prevotella and an increased level of Colonzella is linked to the development and progression of rheumatoid arthritis. Whilst a reduction in the beneficial ones, like Fecale, this one comes back, and Bacteroides also play a role. So can you see it's about balance between good and bad? And although we use the word good and bad, nothing is bad. The lion's not bad. The lion will eat you if you're in the room with it and it's hungry. It's doing exactly what it's meant to do. And that shark will eat you if you're in the ocean because that's exactly what it needs to do. And so these bacteria are just living, they're doing their thing, and they happen to be bad for us when they're in imbalance. But nature's put them there for a reason, and it's to understand the balance is what's important. What about cholesterol? You know, they're studying a concoction of bacteria that can drop total cholesterol by 15 to 20%. That's like the non-statin therapies that are medically sold out there. Certain gut bacteria like Acinetobacter and the Eubacterium can metabolize cholesterol and are associated with lower cholesterol levels, potentially reducing cardiac risk if you believe cholesterol is directly linked to cardiac risk. Of course, there's more to that. There's inflammation as well. So where does this lead us to? So you guys wanted to talk on not only on the gut microbiome, but what can you do? What can you offer? So gut therapeutics is about to be launched to give you an idea of what's coming. This is a 14 strain, and you must understand that all of them have been selected for a reason, having studied this for a long time, looking through many studies. But if you wanted a probiotic to keep you in balance once you sorted your shit out, this at the moment would be the highest probiotic dose with the most variety with a prebiotic currently double to the highest dose in the industry thus far with digestive enzymes and Saccharomyces in 2 capsules. So remember, it's a combination drug. That's why there's so few combination drugs on the market, because it's very difficult to put all these things in a small space, and then to get the right ones chosen and to try and do a lot of things. So this is not to cure. This is to maintain somebody who's had a problem. You fix them up, and it's a nice to have as a maintenance dose. The closest one at the moment comes to 16 billion, and I was involved in developing that about 20 years ago. So that's what you could fit in those days. It did not have the Saccharomyces. The enzyme content was, some of them were half, some of them were about a third to 2/3 of these. And certainly the prebiotics, we could only fit in half those days. So you've come a long way from the single strains, the 5 strain, the 9 strains, 14 strains. So just like that, there's a 250 billion coming out. There's a 500 billion. There's a trillion on its way. There's the lean one with Echinacea, but not only Echinacea. Echinacea is giving you a buzzword now. That's why the whole world's talking about it. But listen, there's a few others that are also, I've just shown you, that are very important for metabolic conditions. So the concoction that's coming out is with over 500 billion, 60 billion Echinacea, with a lot of other ones that I've mentioned that are involved in terms of gut microbiome and weight and metabolism. Gastrogard, what is this guy? So we know colostrum is important because that's why moms give it to you in breast milk. The IgA is protective. Until your immune system gets developed, mom's going to give you immunity for the first 3 months. And so, and then people tell you glutamine is important as well. They are right, to maintain that mucus level. And people are going to tell you another thing is important and another thing. And so we've taken all these other things and we've put it in a supplement to help maintain gut inflammation and that cell integrity to keep the tight junctions closed as opposed to allowing a leaky gut. This will be the first good postbiotic. So you get butyrate. You guys will say, yeah, but butyrate has been on the market for a while. You're right. This one will be butyrate. It's got acetic acid. It's got propionic acid. It's got pre-digested guar gum. It's got lactulose. It's got a lot of fiber. Because remember, the fiber from that slide I showed you is to feed your pets. You want to feed more than one pet, not do what I'm doing every day, eating cabbage, probably feeding a few guys and hoping that their waste product keeps my other guys alive. So this is going to be top range and the first of its kind with basically a blend of all fibers, but fiber not just to form your stool, but to add postbiotics to dampen inflammation, help the metabolic condition, but also maintain good gut microbiome and rigidity of that single cell layer. And then there's one coming out for allergy. And so there's a few in between as well, but I thought, let me mention these and they'll be on your shelves in the next 6 weeks. They're just finishing the labeling and getting it ready for SAPCRA approval. So this part of the talk, I'm contributing towards the tool. So how do these patients present to you? These guys with problems with the gut. It may be bloating, excessive production of gas and inflammation in the gut. Remember, you've got 6 meters of the stuff. And so even if it's a bit inflamed, there's going to be a big increase in that gut abdomen. Then there could be excessive flatulence when there wasn't there. Stools are important. We don't get taught to decipher stools at medical school. And so a stool, you can Google this thing, it's disgusting, but a stool that is like toothpaste, and I've spoiled it for you now, brushing time. Well formed, bulky enough, not stringy, that doesn't break up, that floats. That's a good bowel movement. That's representative of everything a bowel should have. So anything other than that, loose, broken stools, pellets like little bollickies, constipation, diarrhea, bloating, excessive flatulence, cramping, pain, blood in the stools, that's a bit extreme. All signs that the gut microbiome is not too healthy. So what is the tool that we used up to now to work out what's happening with the gut microbiome? It might not be perfect. Okay, some of you guys will tell me, yeah, but you can send stools off. Yeah, you send off your stools at lots of money overseas, and it takes 3, 4, 5 weeks, 6 weeks to come back. And this is a very dynamic tool. So the patients go through a treatment, 6 weeks, 12 weeks, and they come back. Now you have to wait for another stool test. It takes so long for it to come back from overseas. And so we've been doing this now for well over 20 years. So we needed a tool that was much better. And those days, stools were still in the infancy and the costs were high. My patients in Dubai use 1 of 3 labs, and these things cost horrendous amount. I know one in Europe is about 850 pounds, the really good ones. One's around 1,650 euros. The other one is 2,450 euros. And of course, it mustn't become like a fad where you just send off genetic studies like everybody's doing now and then hope somebody interprets it. Because I'm not even asking for it and I'm meant to be interpreting it. And I'm sure you guys are asked to interpret it as well. So I think these things should come with a good report from the guys doing the genetic test with some clinical advice. But I mean, lest that doesn't happen. The point is, it's not about just sending the stool off. What is it that you're going to do with it? So you employed live blood analysis. For those of you that are medically trained out there, remember third year microbiology? Remember you had to sit under the microscope and identify stuff? What do you think you were doing there? You were doing a live blood analysis. The thing is, you were looking for something different to what Kim looks for. So, do you remember when a patient doesn't have spleen? What do you see? You see certain types of cells. You see ruptured red blood cells. When you have a patient who's sitting with a metallic valve in the heart, what do you see? You see something. When you got a patient with bar bodies, dumbbell bar bodies, it tells you something. When you got certain white blood cells looking differently, it starts telling you, oh, maybe this guy's got a leukemia, maybe this guy's got a lymphoma. When you look at a red blood cell that is not looking discoid like a disc, it's premature. It's been pushed out a bit earlier. Maybe there's a problem here with iron, B12, or folate, depending on its size. So iron deficiency will be small, the B12 and folate will be big, and so forth. When you got mosquito, when you got malaria, you'll see certain things. So remember, we only spent a few weeks looking under the microscope to try and understand. You could see uric acid crystals under other fluids. There's a lot more that can be picked up. So these people are trained to do so. So we used to use live blood analysis. Patient drops their blood on the microscope. We got a big screen. They can see what's happening. And basically, it tells you a lot about the bacteria, parasites, and yeast, not so much about viruses. It tells you if the digestion process is going on well and quite a few other things. So this is somebody's live blood analysis. One of our patients under a microscope, Kim took a photograph, and this patient's terrain is acidic, which allows parasitic and bacterial overgrowth. And I swear I got this thing to work earlier. Okay, this was live actually. You can see things moving. So Kim, you think it's because I've got a laser pointer this thing's not working? You just want to unmute. All right. Anyway, this was a beautiful video showing you all these things moving around. It's live, it's living, because it's the patient's. Okay, you know what I'm going to do? I'm going to just try and bring it to the smaller page and take this camera out, give control. No, found. I'm going to stop sharing. Anyway, leave it. Later, I'm going to try while I'm answering the questions to see if I can make this guy work. There we go. And so a lot of times people say, no, this is all bullshit. Oh, we all know the size of red blood cells. We all know the size of white blood cells. We all know the sizes of platelets. So it's very easy to identify them. And then what else is out there in the blood? And from that perspective, this is another one. So this is showing major microbiome imbalance, excess candidal overgrowth. Now, I remember when we used to tell some people you got a candida, they say, that's bullshit, you'd be dead. Why? Because we remember seeing HIV patients with candida dying in ICU. And yet now, the Lancet test blood, urine, and stool for candida. And these are living people walking around, and you can have bacterial overgrowth in the stool, and you can have candida overgrowth in the body as well. We see this now. But here we've seen a lot of undigested fats and a parasite, but the other one shows it a bit better. So let me move. Oh, here we go. Look at this. Scary shit, right? Look at that center cell. Candidal overgrowth. Anyway, so this is showing that the patient is actually not digesting proteins too well, and some parasitic forms as well. This one's also showing a gut parasite. That's Kim's voice, by the way. Anyway, so what's the basis of this? You got a leaky gut. It's more permeable. It's going to be allowing stuff to come through that's not meant to come through. So you can see it under the microscope. The beauty is when you look at it before and after, because we tape this. So the patient can see the before and the after, and they know it's their blood and it's dated, and and then they can match it with the symptoms and they can say, ha, and then there's an aha moment. And so after 20 years of doing it, that's the tool we were using. It's cheap, it's live, it's right there in your rooms. You're getting this answer straight away. And while stools is going to be introducing a big revolutionary change to how we're managing this, I think the combination of the two tests are going to be good, because the stool the stool test is not going to, it's going to give you what they call a shotgun ribosomal look at the different bacteria, but doesn't give you that much more than information in terms of protein and fat digestion. So I think a combination of the two tests are important. So now I'm going to quickly go through some of the dietary things that need to happen as part of what you're managing. So the patients that are intolerant to stuff, and you guys can Google this. Elimination diets are important. Try and work out. The patient needs to work out for you, like they found out that they're allergic to cats by exclusion, or eggs by excluding it out of their diet for a few days and then introducing it, and then they can see what's happening with the bloating or the intolerances or the nausea. Elimination diets are important. And if your patient's very inflamed, you've got wheals, you've got rashes, hives, low histamine diets are important. So you distinguish between high and low histamine diets and see if that improves the patient's gut microbiome. IBS, things you can enjoy and things to avoid. The big ones are obvious: gluten, but sorbitol. You know, we think sugar is bad. Yeah, but the non-nutritive sugar molecules are also very bad from a gut microbiome perspective. Things to avoid. FODMAP, important. This stands for fermentable stuff. So certain short-chain carbohydrates are not absorbed in the gut, small bowel, and then will ferment in the colon and cause issues. Oligosaccharides, disaccharides, important, monosaccharides, and polyols that you can read about. And I did a talk on it last year, but it's important. So we're coming down to now the end, and I think this one puts it nicely. We've got good commensal bacteria that interacts with the pathogen recognition receptors I showed you on the brush border, that then activates the T cells, helper and suppressor cells. So we've got protection against pathogens. The short chain fatty acids from dietary fiber acts as an anti-inflammatory agent, and the T regulatory cell expansion then improves immune homeostasis. So this is to have a good, healthy gut, and a good, healthy gut, as Hippocrates says, means medical conditions are going to stay away. And he said all pathology starts in the gut. This is the father of modern medicine. What about dysbiosis, or leaky gut, as the homeopaths refer to it? Lifestyle is important. They've shown how exercise influences gut microbiome and inflammatory molecules as well. So dietary changes I spoke about, alcohol intake, certain drugs will influence it. So changes in tight junction is going to allow pathogens to get into the circulation. Production of not short but long chain fatty acids is going to lead to inflammation. Inflammatory signals, autoimmunity through genetic mimicry. So that's the difference between a good and a bad gut microbiome, and the majority of these organisms live here. And when there is an overgrowth, in some patients it can go through into the small intestines, which would be sitting there, and cause SIBO, small intestine bacterial overgrowth, and obviously cause problems. So for those of you who need your clients to have a live blood analysis done, and Kim gives you quite a very nice report, a tick chart showing you more or less what's happening with you can get hold of her. She practices 6 days of the week, but half a day on a Monday and half a day on Wednesday she's with us. And you guys can get hold of her there, or you guys all have Leonie's number. They'll put you in touch. Don't call me. There's a WhatsApp, so I'll leave it here a bit longer, and she can help you with live blood analysis unless you have other tools of measuring it. But for now, there's a punt for Kim Samaritano. So I'm going to stop sharing in a little bit, and you guys I can see your questions, but this was sent through this week by a good doctor. Good day, Leonie. Dr. Bana may have already covered some of these questions, but in case he hasn't, please can you add them for me? Thank you, Dr. Singh. What negatively affects your gut microbiome? Modern living. In short, we've touched that, right? From being born the wrong way to not being breastfed to be given. I mean, I'm still looking for the gynae who told the obstetrician who told my mother, stop breastfeeding him and give him cow's milk. That's why I'm so bloody having such a big gut microbiome and allergy issue, right? So foods, they have preservatives, too many antibiotics, yeast. There's lots of things. I think we've covered most of the things that affect the gut microbiome. What are the signs of an unhealthy gut? I've touched on the symptoms of that as well. What I haven't added there, though, is fatigue. Brain fog, fatigue, big. In my practice, it's easy. I say there's 4 big causes of fatigue. Thyroid, adrenal, thyroid, iron is an easy one, and then gut microbiome issues. How do you improve and restore gut microbiome? So it's got to do with lifestyle. It's got to do with eating the right foods. That is pre and postbiotic and maintaining the good bacteria. It's about keeping well hydrated. It's making sure exercise is important, and then making sure that you keep antibiotics at the minimum and watch the preservatives and colorants in your foods. And there's a lot one can do. But the main thing is, I think when this fiber comes out, it's going to be easy. But otherwise, you've got to go to eat lots of different fibers like I do every day now. How can you rebuild your gut microbiome after a course of antibiotics? Typically, we tell clients once you've had a course of antibiotics, you want to be giving an extremely high dose of probiotics. So, okay, so we get a live blood analysis. And then what do we do with this? So based on what we find, the theory that I've always used is something I've borrowed indirectly from my microbiology second and third year lectures doing BSc at WITS. So when we used to try and grow these things in culture, you know, in these little petri dishes, there's something called competitive inhibition. Competitive inhibition. So people always say, but how do you know you're getting the bad guy? Who's the bad guy? I don't give a shit. I'm not trying to look for a patent. So the guy who's looking for a patent, he or she must go and find out that this guy is Prevotella and it causes rheumatoid arthritis. They want a patent and they're going to make lots of money for 16 years for it. So we make some assumptions. What are the assumptions? There's something bad here. I can see it's an overgrowth of Candida, treatment X. I can see there's an overgrowth of bacteria, whether it's L shape or rod shape. This is what we're going to be doing. I see some parasites. I see a combination of those. I see fungal forms. I see mycoplasma. I see whatever. Okay. So we know there's something bad out there. Well, how do we get rid of it? What do you think the elephants are doing in Kruger National Park now? I don't think there's anything as such as overgrowth of elephants. I think we've restricted their space, so they've overgrown their space, and now they're basically breaking all the trees, and that's going to affect the entire biosphere, and that's going to have a downstream effect on basically the buck and the giraffe and then the lions and so forth. So you're messing with the whole microbiome because we restricted them in one area. Now let's use the same analogy here. If you've got an overgrowth of Yeast. Let me take a good bacteria that I know, no matter in what doses I give it, the higher the dose, the better. And I know no matter how much I give, it will never, ever be dangerous to the client. Competitive inhibition. Send lots of the guys on your side of the army, and it's going to kill the bad guy in time. How does it kill the bad guy? It outdoes it. They're fighting for the same environment, same food. And so it's called competitive inhibition. When we used to take the petri dish and we used to swipe a certain bacteria, once it starts overgrowing, it will prevent all the other guys from growing. And so that's the thing I employed then for my third year lectures. And I said, right, modern medicine's not doing anything for my gut because I was first treating myself. And I said, right. So this lady told me lactobacillus, we know it's absolutely good. And so henceforth, we started saying, which are the bacteria that we know 100% from the studies that have been done already that will never cause danger. Put lots of them in. Lots, not 1 billion. And eventually they'll get rid of the bad guy, whoever the bad guy is. So you're basically resetting the gut microbiome. Now you've overgrown the good guy, but eventually that entire thing's reset then. So that's how we worked on it. So that's how you can rebuild the gut microbiome after a course of antibiotics. Put high doses, extremely high doses of a good one. So your interflora, 5 billion, 2 billion ain't going to do the trick. What is the latest research on mental health in microbiome? Microbiome affects mental health. There's work on schizophrenia. There's work on anxiety. And there's definitely work on depression and serotonin levels. In fact, they've shown that the gut microbiome, and this was a publication in New Scientist, I don't know, we left Melrose Arch about 13 years ago. Probably about 11 years ago, there was a publication in New Scientist already showing how the gut microbiome plays a much bigger role on mental health than we had thought. How does diet affect my diet? So I've spoken about that, right? There was a whole slide on fermented foods, probiotics, prebiotics, postbiotics. From Dr. Owen, for the upcoming webinar, I would like to understand the impact of a full right hemicolectomy on the gut microbiome. So there's the left, there's the right, probably had a tumor, and they went and took out all of that. So what's the effect of the full right hemicolectomy on the gut microbiome when the patient had resection, including terminal ileum, cecum, ileocecal valve, ascending colon, and small part of the transverse colon? This patient is severely compromised from surgery as well as 8 months of chemo. They obviously will have issues with absorption of vitamin Bs and zinc, that the patient does not tolerate many foods and has symptoms of the short bowel syndrome. The patient does not tolerate many foods, causes diarrhea, occasionally constipation. Prebiotics are not well tolerated. How can we optimize this patient's quality of life, mitigate small SIBO, small intestinal bacterial overgrowth? There's a lot here. Symptoms of short bowel, not being absorbed in nutrients, the effects of chemotherapy. Right. So there must be a reason why your maker put this valve here. There must be, isn't it? What the hell? I keep going on the wrong side. So the valve's here. And then you get the small intestines, and then you go higher up in the stomach, and then you've got the esophagus there. So what we're hearing is that valve, terminal ileum, is a small part of the small intestine, and all of this, and half of that is gone. Now, obviously, that's going to affect the gut microbiome. You don't have to be a rocket scientist to know that, so I'm not going to get prizes for that answer. But the important thing here is that you had chemo, the patient had chemo, so that in itself, whether it's chemo or radiation, affect the entire gut microbiome. So we need to now try and recolonize this bacteria in this part as best as we can, because if you get the bad ones here, it's already overgrowing, and this thing's been tied to the small intestine, and there's now bacterial overshoot or no retrograde movement of it into the small intestines and causing SIBO. So lots of things to unpack here. Not an easy climb. So here what I would say is that wipe the slate clean. Number one. And the chemo may have done that, but after the chemo, a lot may have happened. Wipe the slate clean and then recolonize the gut with good microbiome. The selection of those microbiomes is going to be important. Go for as high a dose as possible. Keeping in mind, and this goes for all patients, when you start probiotics, you'll see on the bottles or somewhere in the insert, they'll tell the client in the first few days or in the first week or two, the gut may get worse before it gets better. Same thing with digestive enzymes. So here I'm not saying you need digestive enzymes. I'm just saying here, probiotics, prebiotics to keep the good guys alive. And then from a SIBO perspective, here you may or may not need help with the bacterial overload. So here, it'd be nice to do a live blood analysis or send this patient stool sample and then try and work out what the overgrowth is. Because for some bacterial overgrowth, you do need antibiotics initially, but then you need to come in again and recolonize the gut with good bacteria. So this one's difficult, but certainly for the symptoms of the small bowel, introducing short chain free fatty acids will be important for the inflammation, good bacteria, and then you may or may not need digestive enzymes based on what the stool consistency is like. So I'm going to now, let me just see if I'm finished. Okay, this is Leonie's advert for the next talk. We're going to be talking about oils. Are all oils equal? What's the difference? And then you guys all know how to get your CPD points from Leonie. This is the website for the CPD certificates. And then any information that you guys need from Leonie. So I'll leave this now. So let's see. I hope there's a lot of questions. There's still 63 people that are live. And who's got a big attention span. Let's see what's happening here. All right, questions. Okay, so the CPD point link is here. I cannot see the slides. Who is presenting? Oh, that's a bit. I hope you guys could see it. Okay. Is it possible that the younger gut is not fully developed and that's why it can't be tested on children? Sorry, I missed that. What can't be tested is in the gut microbiome is not as diverse as yet. Okay, so a young child's gut can be tested. You can test the stools and you can get a shotgun microsomal test on that. In terms of it's not yet fully mature, that's what we mean, right? And also, whilst you know the gut of this child has been colonized already, you don't know the genetic influence that's coming along with this gut microbiome. So that's what we mean in terms of why we were not using, why we don't want to use a young child's stool to be inoculated for something chronic. For C. diff, it would be fine. Do you ever see a healthy live blood? Yeah. Darlene. So one patient told me, I mean, this looks like bullshit. Like it's like vitamin D. Everybody's vitamin D deficient. I tell you, there are people with good live blood analysis, and yeah, it is amazing. But we've had a few where they're sitting with very good live blood analysis from the onset. Of course, once we work on it, we can show them a before and after. And some patients' gut microbiome improves, and others like mine, you always just got to manage the problem and try and maintain it. I don't think you ever get over salmonella. What are the laboratory investigations to look out for as poor indicated for gut health? Nothing. That's the problem. The lab's not going to be able to show you shit. Mind the pun there. Okay. So, of course, the gastroenterologists use a test, but that's like for gluten insensitivity. That's like quite late into the game already, and that's only for gluten. Um, and you got some tests for celiac disease. But just in terms of irritable bowel syndrome, that's why it's always been a dustbin diagnosis. I mean, how do gastroenterologists make that? They put the scope in, there's no ulcer, there's no cancer. They put the scope the other way. There's no diverticulitis. There's no Crohn. There's no inflammation. So they're saying, right, and there's no bloody patches and it. So there's no Crohn's. There's no ulcerative colitis, and there's no cancers. Then there's no polyps. But the patient still have symptoms. So they called it irritable bowel syndrome. That's been like a 2.5 decade dustbin diagnosis. And then what happened was it evolved. The Romans came up with a criteria, and they said they divided it into saying, all right, is this predominantly diarrhea? Is this IBS predominantly constipation? Is it mixed and so forth? And they tried to start differentiating it, but there was never a test. And so that's one of the issues. If there was a test, sure, we would have been much further ahead with can LPS be measured in labs, and is there any merit in doing it? No. I missed your reply to Dr. Singh's question 2. What are the signs of an unhealthy gut other than gut symptoms? Fatigue, brain fog, bloating, which is the obvious one, excessive flatulence, loose broken stools, abdominal pain, which you may or may not have, and yeah, you guys know most of them. And one last one, of course, blood in the stools that is not from anal fissure is something that you need to go and look for in the gut, whether it's a cancer or whatever. How about SIBO, which is so common in IBS? Giving them probiotics may make them worse. Yeah, that's why I said, Talin, SIBO is a lot more complicated, and that's why I said it's important for you to make the diagnosis of SIBO properly, because that's where I said the client may initially need antibiotics. And you'd like to know whether they rod shape or at least cone shape if you can't identify which particular one. So that becomes a lot more complicated to manage without tools. How would you help a patient who has been on chronic use of PPI? Send them to the next doctor. No, I'm joking. Okay, so this is a very tough one now. So let me give you a personal story. I do believe the maker's always given me a condition to learn something to move me hopefully in the right direction, and then maybe to help one or two other patients as well. So my wife's been trying to get me to go for a colonoscopy for years and years and years. My father's survived colon cancer, and my maternal grandfather also had a bag. So, of course, I've got a tattoo saying I'm at risk of bowel cancer. So, of course, you're meant to be going for this between 45 and 50. Needless to say, I'm 58 now. But when I was 56, the directors at CAM and a few directors at Discovery decided we'll have it all done at our day clinic on a Saturday. And I was just forced to do it because Jeff wanted to do it. And he says, right, all of you guys are going to do it. So my wife was very happy. And so, I mean, just the preparation. We put a man on the moon. You'd think the gastrologist would have come up with something better to clean your bowels with. I don't know if you guys have gone through it. It is so archaic. But anyway, that was the thing that was putting me off this whole thing. But anyway, we did it. And lo and behold, this gastroenterologist comes straight to me first, and he says, yours was the worst. So I said, what do you mean? And so, okay, so let me track back. So this was in November, about February of that year, I ruptured my L5S1 disc. Prior to that, I was exercising and doing karate like a demon. So I was fit. And in any case, now I stopped all activity and my weight hasn't changed for years and years and years and years. Anyway, I put on 2 kilos. And so every time I bend, I get a bit of reflux. I think, okay, no shit, it must be this extra 2 kilos, putting a bit of pressure, not exercising, whatever. Anyway, he said when they put that pregnancy looking test and they put a fluid on it, it never ever turned pink faster on any other patient they've ever seen. And then he turned to his nurse and said, can you tell Dr. Bana this thing has never turned pink so quickly? Okay, so that means I had Helicobacter pylori. So what do you think they do? They give you bloody triple antibiotics. So I've already got a messed up gut microbiome. Anyway, I said, no, listen, this doesn't sound good. Let me just take it. So I'm on this shit for 28 days. Whole and behold, that reflux goes away. I still got the 2 kilos because I still can't exercise. I went for my op 2 weeks ago. I had microsurgery. If any of you guys need a good neurosurgeon, the best in the world, microsurgery, I walked out of the hospital 3 hours after the op, went home. He's the best neurosurgeon in the country. Microsurgery. Half my disc was taken off. So anyway, and so, hey, Noel Zaidi is there. He's the one who told me I got Helicobacter pylori. Anyway, so Dr. Zaidi gave me this treatment and I took it and my reflux disappeared. Whole and behold, I said, wow, I am sorted. Come February, all those bloody symptoms are back. I said, no, I'm not going to go back to Noel's ID. This thing is back. I said, no, let me start using my own medicine. That's when I started eating my goddamn salad every day for lunch. If you guys have a look at it ever, it's like a big tub like this. 80% cabbage because it doesn't bloat me. Of course, it bloats, you don't do it. Find another fiber, and then there's a bit of evo, and then there's a cucumber, and there's pepper, and all this, and few nuts. And so it almost looks like all the colors every dietitian would be very happy with. It's got all the food groups. I do that every day, and whole and behold, 6 weeks later, all my reflux symptoms are gone. Why? Because I'm giving the system good prebiotics to feed the good guys. Let them outdo the bad guy. And since then, all my symptoms are gone. And my Helicobacter antibody test on my blood is negative. Anyway, it can come back again. What I'm saying is that, and we'll ask Dr. Zaidi to maybe answer that, long-term PPI use can't be good if you're switching off the acid forever. Can't be good for the bacterium lower down because you're allowing recolonization with more bad ones. But we'll ask Dr. Noel Zaidi. Dr. Zaidi, do you want to come on and maybe tackle that one? Okay, maybe later. Could you reverse lifestyle-induced seronegative rheumatoid arthritis? Could you reverse lifestyle-induced? Sure. Tough one, huh? When you're sitting with autoimmune conditions, I tell patients it is tough. We've had a few patients going to remission with ITP, certainly with asthma, early cases of Hashimoto's, where, and I presented some of these in last year's webinar with the antibodies going negative in some patients. But to consistently do it, I'm saying we're still early. I've shown you Pervitella's role and Bacter's role in it. So I think things are changing now that we are identifying the bacteria that cause it. But I'm sure in the future, we're going to be having a lot better results. Good evening, Dr. Bana. Are there any specific probiotics brands that you would recommend when starting treatment for any autoimmune conditions? Okay, so Dr. Brink, you know, for years and years and years, I've been using different ranges of probiotics. Like I told you, the concept of competitive inhibition is go and give the highest dose of known bacteria, try and get it to be as diverse as possible. That will outdo the bad guy. In the meantime, if you need colostrum to help the inflammation of the lining, it will be important. Sometimes you can use glutamine to help that as well. And then it's about maintenance and then making sure that the gut is right. try and avoid antibiotics in the early phase of the treatment unless you have to. And like Darlene said, SIBO is something else. And remember that everything on the whole again range, if you've been to some of the other webinars, out of need. When there's something not in the market, then it's come in not to compete against others, to uh, come in and offer a tool that hasn't been there before. That's why you'll see the kind of numbers we're looking at here, 250 billion, 550 billion, a trillion. That's why there's the first Ecomensah product of its kind in the world, with not only Ecomensah at 60 billion, but there's something else making up the other 460 billion. It's about 520 in terms of some of the other bacteria as well. And so, yes, there's a company called Endobiosis that's doing stool analysis. And I'm actually having a meeting, I think on the 1st of April, with the CEO. And I just want them to see if they can modify a few things on the report. But yeah, soon we're going to be having stool analysis being done much more easily in South Africa. And so in terms of brand names, check what probiotics they got. That's why I tried to innuate not all probiotics are the same. Now you at least know some classes are good, some classes are bad. What's good for rheumatoid arthritis might not be the same for diabetes, what might not be the same for obesity, and so forth. So individualize the treatments. But yeah, numbers are what's important. Now I'm having difficult. Oh, Dr. No,
Here
do
I
you
am.
want to answer that earlier question?
Remind me of the question. I know you were talking about the concerns
Dr.
that you had about long-term PPI treatment.
Yeah. So somebody asked, how do you get somebody off who's been on long-term PPIs? As you know, some people are on like 40 milligrams of Trustin twice a day for the last 4 years. What do you do?
Yeah, I think the recommendations that I can give is that one is able to reduce their PPIs. One has to do it slowly. In other words, if they're taking, let's say, for instance, a double dose of PPIs, I like to reduce, first of all, reduce the milligram dosage slowly over about a period of a month or maybe even 6 weeks, and then reduce that further, then go on to alternate day therapy, then maybe twice a week. And at some point in time along the way, you may get to a point where they come back to me and say, I'm beginning to suffer again. And it gives me a threshold to work on. Some people can get off the PPIs and stay off for a few months, and then they may have to come back. But I think, the crux of the issue is that one needs to reduce that dose slowly. That's number one.
Okay, thank you.
Okay. And then just a quick question about the acid reduction that one expects. If you take a maintenance dose of a PPI, you only reduce the gastric acid production about 15 to 20%. So it's not as though you're switching off all the acid. So in terms of protein digestion, that there's not much effect taking a maintenance dose of a PPI. I'd be more concerned about giving a patient a PPI who's got osteoporosis. As we know, that potentially long term, the PPI can affect the osteoporosis and aggravate the problem.
Yeah. All right, thanks. So in terms of the protein digestion issues that we can pick up on live blood analysis what's happening with the protein. And so you see these macromolecules that haven't been digested, because remember, you only meant to be absorbing protein in an amino acid state, or two amino acids bound together, not more than that. Then it becomes macrogenic. And when it's when it's a macromolecule, it becomes more enterogenic. And then you have enterogenic presenting cells, and then you can develop intolerances. And that's where issues start. So from that perspective, that's why we need to find other ways of getting them to digest the proteins if the acid is not there. And you might be aware that some people actually use betaine hydrochloric acid in states of achlorhydria to actually improve that. So that's where you guys can come in and help us to see what the pH levels are with your monometry tests.
Yeah, so those were tests that we in fact did several years ago, but we used to take gastric secretions, and we used to evaluate them and see what the gastric pH was. But I mean, I can tell you in the past 20 odd years, that's not something that we're doing anymore. For some reason, it went out of vogue, and we've never really gone back there. So
Yeah.
I'm not sure. I'm not sure why that went out of favor, but we haven't been doing it for years.
Yeah, so when patients ask me, should I be using betaine hydrochloric acid? And I tell them, well, you can do the poor man's test. And the poor man's test is you buy a bottle and you
Yes,
take it before a meal. And if it burns, you didn't need it. If it doesn't burn and the bloating improves after your meal, because if you took it because you were bloating after a meal that was high in protein, then you know you need it. And it's
sure.
mainly because I wanted you to answer that because I know most of our colleagues are basically not doing that test.
Yeah.
Whereas in the past, it was actually easy to work out who had a low acid production in the body as to high. Okay.
Yeah, and I can't give you an answer why those tests are not being done anymore, but they've just really gone out of favor for
Yeah.
whatever reason.
All right. All right. So, yeah, we're hoping that the gut microbiome tests are going to be done cheaper in South Africa. I know more or less the price, but just give it time. Maybe on the next talk, I'll give 5 minutes towards that and tell you guys more about the test that's coming in South Africa. But more importantly, hopefully it has a report that can help you rather than just telling you which probiotic to use, because they got an AI tool that helps you with that. And the doctor who asked, it's much cheaper than sending it to Japan, much cheaper than sending it to Europe. And the future of this thing is evolving at a very fast rate. So just like you're going to be able to read your entire genome for $100 in the next 5 years, at the moment it's around $500. It used to be 2 years ago $1,000. And this is something that took 5 billion and 20 years to measure. You'll now have it in a very short period of time. That same technology allows us to use shotgun ribosomal technology to now work out what's happening with these gut microbiome. And the more tests that are done should bring the cost down. So let's see what happens. All right. So I hope that was helpful. And if you got any questions, leave it with Leonie. I'll try and answer them as best as I can or refer them to somebody who can. But thank you guys, and I hope it's been helpful. Thank you and good night.
Thank you.